Hi, my name is Lela Legesse, and here is my master's thesis project. Did you know that we have about 100 trillion cells in our body and over 200 different cell types? But have you ever wondered what causes these differences in cell types? For instance, how do our brain cells, blood cells, or even our muscle cells develop? Let's take a look at what a basic cell looks like. Inside a cell, you have structures called organelles, and each organelle has different functions. For example, the nucleus is where we store our genetic material, and in the mitochondria is where energy is produced. And the endoplasmic reticulum, also known as the ER, is where proteins are made. These are just some of the important parts that make up a cell. As you know, cells are always dividing. From the moment of conception, when the sperm fertilizes the egg and a fetus is developed, all the way up until the moment we die. There are two different ways a cell can divide. The first way, which we all are familiar with, is when a cell divides and makes two identical copies of itself. This is what we call symmetric cell division. The other way a cell can divide is when you begin with one cell and instead of two identical copies of itself, it will make two different types of cells. This process is called asymmetric cell division. A few years ago, the Riggs lab at San Francisco State University discovered that the endoplasmic reticulum actually divides asymmetrically. They also discovered that in order for this to occur, a specific protein called jaganol is needed. This finding provided insight on how organelles such as the ER are inherited and how this plays a role in cell diversity. Let's go a little bit deeper into these processes and learn about how exactly a cell divides asymmetrically or symmetrically. One important process that has been studied and known to be vital to asymmetric cell division is cell polarity. So what is cell polarity and why is it even important? Cell polarity is simply a cell's ability to sense and determine direction. You can imagine a cell needs to be able to distinguish its front from its back, its top from its bottom, and its inside from its outside. One of the best ways to study processes such as polarity and asymmetric cell division is by using a model organism called Drosophila melanogaster. You may know this as the fruit fly. This organism has been used for many genetic and molecular studies. The fruit fly is simple to work with. They are very small and thus very easy to maintain and produce in large numbers. They have a simple genome with over 60% of genes involved in human genetic disease. They also have a relatively short life cycle. They can go from embryos to first, second, and instar larvae to pupa, and then adult flies in about 12 days. In my lab, we focus mainly on embryonic and larval stem cells in Drosophila, specifically neuroblasts, which are the stem cells found in the brain that will eventually develop into the central nervous system. So how does asymmetric cell division in Drosophila neuroblasts work? In the central nervous system, embryonic neuroblasts undergo asymmetric cell division along what we call an apical basal axis, which is just top or bottom, and that produces two daughter cells, a self-renewing neuroblast and a GMC, also known as ganglion mother cell, that go on to produce neurons and glial cells. There are key proteins and protein complexes needed that are unequally positioned at either the apical or basal end, which cause the neuroblast to divide asymmetrically. This further led me to asking my particular research questions, which is, does ER asymmetry during mitosis play a role in establishing cell polarity? I wanted to know what comes first. Does the ER dividing asymmetrically cause cell polarity, or does having cell polarity cause ER asymmetry? So to answer this question, I developed two aims, which were focused on examining the role of some of the proteins that are found on the apical side of the neuroblast and to see if they are responsible for establishing ER asymmetry. My first aim was to determine if the protein jaganol was found at the same place as the apical proteins in the neuroblast. My second aim is to use fly mutants, which are just flies that do not have these particular proteins, and examine the ER to see if the asymmetry is affected. I will be using molecular techniques such as immunohistochemistry, where I will grow and collect the appropriate stage larvae, dissect the brains, fix and stain them using my antibody of choice, and then image the slides.